Synthesis of glycopeptides is impeded by the high cost of glycoamino acids, their rarity and lability. While traditional resin-based peptide synthesis methods are useful for synthesis at micromole scale, methods to generate targeted libraries at sub-micromole scale did not exist. To help address this, we developed Glyco-SPOT synthesis method, which can be used to synthesize libraries of glycopeptides in nanomole or microgram quantities in a cost-effective manner and without the use of expensive equipment. This method builds on spot synthesis technique, which utilizes cellulose filter paper, and the synthesis is performed by spotting the reaction solutions on the paper. Coupling of glycoamino acids often requires longer reaction times, making them more prone to side reactions and lowered efficiency with the open nature of classical spot synthesis. To enable longer reaction times and improve the efficiency of the synthesis, we introduce various modifications to the method termed Glyco-SPOT, which enable longer reaction times required for coupling glycoamino acids and minimize side reactions. Repeating the synthesis cycles, the peptide sequence is built on the surface of the paper. To test the efficiency of the reaction and ensure completeness, we developed a biopsy test to sample the surface of the paper. After the complete glycopeptide sequence is built, the peptides are deprotected, released from the surface, and purified to produce the glycopeptide libraries. In this paper, we describe the synthesis of two libraries using this technique, encompassing several peptide and glycan sequences with a variety of linkages and including non-natural amino acids as well as labels such as fluorescein. We demonstrate that microgram scale glycopeptide libraries are sufficient for a variety of applications, such as in microarray experiments to test the binding of various glycan binding proteins such as lectins and antibodies, in screening enzyme activity on such microarrays, as well as expanding these microarrays directly for exploring biological activities. To test enzymes in solution for library expansion and enzyme specificity characterization, to measure binding affinity using fluorescence polarization assays with glycopeptides labeled with a fluorophore such as fluorescein, and potential use for mass spectrometry as standards in the future.